plant hormones that, they excrete it, and they're not any use for the microbe, but the plant will take them up. And it can be very beneficial for the plant to have access to a little bit of boost of the hormones or some growth factors, amino acids, vitamins. So we know that there's those dynamics going on. They can also act directly in that they can help fight off pathogens, so they can act as biocontrol agents. We now know that they're capable of ramping up the plant's immune response. So if the bacteria or microbes, fungi, bacteria are hanging out with the plant, they've even shown that, the ones that are hanging out in the soil right next to the roots, so they don't have to be like within the plant like an endophyte. They can just be hanging out right near the roots, and they can stimulate the defense mechanisms and the immune system of the plant. I'll explain a little bit more about that. They can also influence what kinds of chemicals the plant makes. So this kind of goes hand in hand with the plant defense idea, because very often plants will make these bioactive compounds or secondary metabolites in response to attack. So if a plant senses they're getting attacked, they'll ramp up certain key secondary metabolites specific to trying to kill or inhibit that particular pathogen. So plants are sophisticated enough to even know what's attacking them, and then to make the right chemicals to be effective, hopefully, against that particular attacker. We now know that microbes and hanging out on the plant, in the soil near the plant, in the plant, they're capable of influencing what kinds of secondary metabolites the plants are making, and that can help them defend themselves. Certainly, we've talked about how microbes already can help against abiotic stress. We looked at that idea of a virus and a fungus in a plant that can, if the plant doesn't have the virus and the fungus, it can't handle high heat. So we talked about that example a bit ago. This is the idea of not just handling heat tolerance, but dealing with drought, flooding, cold, all kinds of different abiotic stresses. We've already talked about this too, the mineral nutrient availability, that you have to have microbes hanging out in the soil with the plant so that they can get access to some key nutrients, as well as breaking down, just recycling organic matter and releasing those compounds back into their simpler forms, getting things soluble so the plant has access to it. We've already talked about nitrogen fixing microbes. Certainly getting access to phosphorus is a big one. Plants have a really tough time with that themselves. They really do need the microbes, both bacteria and fungi, to help out with that, as well as iron. Iron can be tricky to get access to because many forms of iron are also very insoluble, and it's hard for the plant to get access to it. So they are realizing that this is very much a team effort between the plants and their associated microbes. They're starting to look more carefully at this with the idea of, how can we understand this and then use it to our advantage when we're trying to grow crops for food production for our ever increasing population on this planet. And we need more nutrient dense foods, better quality foods, as well as better yield. And so different companies are popping up now that are doing the work of, okay, well, let's see if we can find some microbes that will fit our needs. Corn, I think like we talked about this last week, corn is a hog for nitrogen. It is very hungry for that. When you grow corn over its life cycle, it does need a lot of nitrogen to finish its life cycle all the way to corn on the cob that we can dig our teeth into. So they're looking at, this idea to create self-fertilizing crops. So corn plants they don't have bacteria hanging out in their roots and forming nodules like with nitrogen fixing situations we've already talked about. They can't do that, but they're trying to do the next best thing, which is this company, what they're thinking of doing is let's get some free living nitrogen fixing microbes in the soil on the corn roots, and then let's get them activated. So the thought is that because when you grow corn in cornfields year after year after year, there's been so much synthetic nitrogen added into the soil that it creates a situation where the microbes that are hanging out in that soil that do nitrogen fixation, they're sitting there going, well, I got all this nitrogen available to me. I'm not going to fix nitrogen because I have it all being fed to me by the grower. And so they turn those genes off because it's intensive. It's intensive to do nitrogen fixation. It requires energy and resources. If you don't need to do that, don't. However, if you can manipulate it where you kind of create a selective force, selective pressure, where you're like, okay, you got to do it on your own. If you ramp back on your nitrogen fertilization that you put into the field and stimulate these bacteria to go ahead and come on, turn those genes on, let's do, get going, get off the couch and start doing nitrogen fixation, then that could be a really good deal because you could use less nitrogen fertilization. That's not super sustainable, even making all that nitrogen fertilizers. And so they want to reawaken this function that has been there all along. And so that's a really smart strategy, understanding the microbe well enough to know, yes, they can do nitrogen fixation. They're just choosing not to because they don't have to. So let's turn the table and make them do it. And so it's really kind of a simple idea. But there's a lot to it. The soil environment is very complex. Getting that balance of turning that on, making sure they make enough nitrogen so the corn can go to completion of its life cycle, so it's not as straightforward as I'm making it out to be, but the big picture idea is actually quite nice. It's a good idea. They just need to get it troubleshooted, I guess, and figure it all out. So this company is looking at doing that. They're looking at, can we get enough microbes into the soil that we know can fix nitrogen, and have them actively do that and not just turn it off and not do that part of their gene expression. So you can read through this as you're studying, but it's a really, really good idea. Another company called Sound Agriculture, and they're looking at, again, microbes. And, the intro here is talking about the sustainability of creating inorganic fertilizers. We have a lot of issues that we're facing with climate change and growing enough food under those conditions, water resources. And so they're really looking at how can we better understand this plant microbe interaction and really use the microbes to really ramp up the plant's health and get them to completion of their life cycle so we can harvest that edible food. They're looking at conversations. The resulting products guide plant and microbe conversations. So what do you think they might be pinpointing when they talk about conversations?
Quorum sensing.
Yep, quorum sensing. They're looking at quorum sensing. So it's like, yes. So they're finally realizing that they could use this information to really ramp up a good team effort between the plant and the microbes, the beneficial microbes. And so they're having some good success. I looked at some of their case studies early on, and they're having some good results so far with some increased yields in fields where they can tweak this versus control fields. So it looks like they're on their way. I mean, this is a company that popped up. They're trying to make a go of it. So there's more and more companies like this. Here's another one by Dr. Linda Kinkle. She was my postdoc advisor at the University of Minnesota, and she was just a powerhouse scientist. She was pulling in multi-million-dollar grants every year, and it was quite exciting to work in her lab. She had a lot of different projects trying to understand plant microbe interactions at a more fine scale. And we had really good success in her lab. The 3 years I was there, we had really good field research results where we could show the microbes were doing their thing and protecting their plants against, primarily we were studying potato scab disease. And over the years, she's been teaching there for many years, and she got frustrated by, she have all these wonderful results and publish this information in scientific journals, but it really wouldn't translate into the real world where real growers were using this information to improve their crop production. And so she decided to start her own company just a few years ago and shifted into this Jord Bioscience. And she just really strongly believes that a couple decades of her work, it needs to get out there. And I just think that's wonderful that she's bridging that gap between, what we do in academia and then getting it out there in the real world. So she's also looking at, let's see, does it say it? So here she's looking at protecting crops. So she's looking at biocontrol agents, stimulating yield. So she's looking at PGPMs for that, and feeding growth. So she's looking at the idea of using microbes to, just make the nutrients in the soil more available to the plants. So she's got all kinds of microbes that when I was there, we isolated hundreds of microbes from soil systems that were they seemed to be very beneficial, particularly against potato scab disease. And so Dr. Kinko hopes her experience will inspire other researchers to increase the impact of their academic work through commercialization activities. And so I think this is great. That's really what we want to see. We do all this hard work in the lab, publish it, but the next step is, okay, now get out there and use it. Let's apply it. And so lots of companies, including hers, that are really starting to make this transition from, okay, we've been learning all this knowledge, let's put it to practice. So you already know that microbes are everywhere on the plant, in the plant. They're very well colonized. It's very analogous to us, how well colonized we are as humans and animals. And so this idea really isn't too shocking that they're going to have some co-evolutionary processes going on where they are benefiting each other so that they can survive and persist. This is from actually my plant physiology textbook that I teach from, and it's showing a few things on here. Again, looking at this idea that the plant and the beneficial microbes, in this case, they're calling them plant growth promoting rhizobacteria, so implying they're on the root area. And so they're showing this bidirectional impact on the fact that roots are very leaky, they exude out certain chemicals, and we now know that plants aren't just like, yes, there's a lot of leakiness to roots, so to speak, but plants are also directly impacting selective microbes. So they have the ability to foster certain groups of microbes over others and attract them to their roots. They have the ability to inhibit others. And so that's what they're trying to get across here, that the plant is much more dynamic than we thought a couple decades ago. They're not just these passive beings that have all this stuff happen to them. They're able to orchestrate certain situations, who is hanging out with them and who is being inhibited. So here you can see that they can stimulate the good bacteria, which then can produce antibiotics to inhibit the pathogens. You have the back and forth exchange of nutrients between the plant and the beneficial microbes. Certainly they're showing the quorum sensing idea, cooperation, as well as competition. So very dynamic between the plant and its associated microbes. They've even shown insects, of course, can cause a lot of damage to plants. And they've even shown that which microbes are present on the leaf or in the leaf, for example. Some of the studies are leaf feeding studies, and they can show that the insect feeding behavior can shift. They are showing that, at least some of the studies that have been published are showing that these insects are responding to chemicals that the microbes are making, and it's making the insect not want to keep eating and move on. And so there's these types of ideas. There's also pollination. Pollination is huge for the plant. To be able to get pollinated, you want your flowers to have the pollen come to them. And I'm going to talk a little bit more about pollination, but basically they're finding that the microbes that are on the flower, in the flower, are influencing this dynamic. They've even found microbes in the pollen grains and on the pollen grains. So there's endophytes within pollen as well as epiphytes on the pollen grain. And so there's trying to get a feel for what's going on there. This is very new research. It's something that has been way overlooked and not really thought about. But they do know they've gone more studies are published on nectar, the microbes hanging out in the nectar, and how they can influence which pollinators are coming and taking the nectar and which ones seem to maybe not like the presence of those microbes and what they're doing in the nectar. So there's this idea, because we know that plants generally want certain pollinators to come because they are well designed for their flower shape to deliver the pollen correctly so they can get pollinated very nicely. And so generally plants have certain key pollinators that they want to attract, and others are not so much desired. And so they're really looking at how microbes might be influencing that dynamic. I'll talk a little bit more about that. They even know the phenology is a fancy word for looking at the timing of the life cycle of the plant. So for example, when does it flower? And they're finding that microbes can influence the timing of that. There's been a lot of effort looking at especially the timing of flowering, and they can show that microbes are influencing that. Epigenetic plasticity of the plant. I'll talk a little bit about that. That's a wild idea, but it's amazing. So I'll come back to that in a second. And then we already talked about the idea of mycorrhizal fungi being there right from the get-go, as you had the shift of algae washing offshore onto terrestrial habitats, and that huge shift from ocean to land is major, and that the mycorrhizal fungi were there to help right from the get-go, hundreds of millions of years ago. And so you can't really deny the fact that plants really do depend on microbes for many, many reasons. This was an interesting paper that was published a couple of years ago, and looking at the microbes hanging out within the flowers and how they impact the pollination. Pollinator attraction, just looking at what kinds of organisms are coming to the flower. And they're showing that microbes influence that. A lot of study with yeasts in particular. So they know that yeasts love to hang out in flowers, especially in the nectar. And so there's understandably a lot of effort going toward all these different yeasts and how they might impact that. Pollinator visits, so they're looking at, how many times, who's coming. And then they have this idea of pollen transmitted endophytic microbes. So now they're recognizing that pollen is colonized both on the surface and also within. So we talked about seeds getting colonized within the seed tissue as it's forming. Now they're looking at pollen. So this is a relatively new field of study, but from this study, here are some of the conclusions and things they were talking about. They certainly have been able to identify all kinds of microbes, not just the yeast, and that you can get them on and in the flower. And endophytes are within the seeds and the pollen and the ovules. And so understanding who's there, what are they doing, how are they impacting pollination and fertilization is a pretty active area. Obviously, so much of our food production, if you think about it, we depend on pollinators effectively doing their job so that that flower can morph into the fruit that we end up eating. So it's a major concern for adequate food production. Okay, the idea of epigenetics is a wild one. This is the idea of you're not messing with the DNA sequence, you're messing with what sequences get turned on and off through a variety of different mechanisms. Methylating the DNA, adding a methyl group, CH3, is a common strategy. When epigenetics first came out, it wasn't actually that long ago. Maybe 20 some years ago, 25 years ago is when this was discovered. And actually, we have the plant world to thank for that. They discovered it first in plants. It didn't make much of a splash at that time because plants aren't humans, but then they found out that it's happening in humans as well. And then it started to hit the news more broadly, but it was actually first discovered in plants. And when it was first discovered, it was quite the saga. A lot of scientists were saying, no, this isn't happening. There was a lot of disbelief and pushback on it. So it required a lot of careful science to persuade that yes, the data is really showing this. So basically, amazingly, they're showing that microbes hanging out in the soil with the plant can influence the plant's epigenetics. And so at the level of the DNA, the plant DNA, methylation, in this paper, they were looking at the methylation of key sections of DNA is influenced by the presence of certain microbes in the soil. So they're not even endophytic into the plant. And so what they were looking at is growth promotion, all right? So they were looking at long-term plant growth promotion. And the other thing here too, it says we found the rhizosphere microbiome assembly was mainly driven by plant development. So basically they're saying the plant was actively recruiting certain microbes over others. So our results showed PGP-B induced DNA methylation in the roots, mediated the growth promotion process. So this is, it's hard, I don't know about you, but it's hard for me to wrap my head around this, that there's that, at that level of the, all the way to the DNA, the microbes in the soil are influencing the plant DNA in that manner. So lots of exciting things to think about with DNA methylation. Is this the one where they showed? Here, this is on the next slide, I think. So in this study, what they did was they had two different strains. PGP5 is a strain of, I think that was a bacillus. Yeah, it was a bacillus species, which you've worked with bacillus in lab. And then PGP41, that's an Arthrobacter bacteria. And so they inoculated these into the soil along with their plant. And 3 days later, they could detect these bacteria. But then shortly after that, they were unable to detect these bacteria anymore. So failed to thrive. So usually when you talk about inoculating microbes for either biocontrol or growth promotion of the plant, the thought is we want those microbes to persist in the soil so that they're always there and actively helping the plant one way or the other. So this initially sounds like, oh, well, that's not the greatest result if after day 3 they can't even detect them anymore. But the weird thing is that they still saw that DNA methylation occurred when they did inoculate those plant bacteria to the plants, and they remained functional after elimination of the microbes. So even after day 3, when they couldn't detect the microbes anymore, they still had an influence. And so important insights into the mechanism also offer strategies for plant microbiome engineering beyond the perspective of maintaining inoculum persistence. So they're saying maybe we don't have to worry so much about that. We should look more into well, in this particular idea, they were looking at the fine detail of plant gene expression and the impact those microbes had. So just essentially 3 days of exposure, and the plant was able to respond to that, shift its methylation patterns. It did stimulate growth because of that epigenetic effect. So pretty amazing study. This was their graphic trying to show this idea visually. So they're showing this idea of we inoculated with plant growth promoting bacteria into the soil. They didn't persist, so they were eliminated. But even in that short time frame, they were able to methylate, or they didn't methylate, but the plant methylated its DNA in key areas that allowed for promotion of plant growth. So that's their hypothesis. They need to do more studies on that to confirm, but the data was good data. It was well done. And a very interesting aspect of maybe we don't have to worry quite as much about getting those microbes to persist. That's a very challenging idea, by the way. Soil is incredibly complex. Billions of microbes in the soil, especially by the roots. And to inoculate your desired microbes into that and then say, please survive this, it is challenging. And they don't always survive. But here it's very hopeful to see that they're saying, well, even if they don't persist long term, they can still have an impact all the way to the level of plant DNA gene expression. So really cool. Here's another wild one. This was published a while ago, but so this is really, really interesting because it's showing microbes in the soil influencing what the plants is doing in the leaves. And so let's walk through this a little bit. What they're doing, they've numbered it. I don't know if you can see. So number 1 is here with the red Pseudomonas. So Pseudomonas syringae is a nasty plant pathogen. And so what the story is here is that they can hang out on the leaves and they can enter into the stomata and start causing problems. This is, I believe, a tomato plant. Pseudomonas love tomatoes. They just love them. And so here's the pathogen starting to do its nastiness. Well, then what they show is that, where's number 2? Oh, here, into the stomata. And then number 3, what they're showing is the plant releases the malic acid from their roots. And what that does is it starts to stimulate Bacillus subtilis. Now, Bacillus subtilis is a good bacteria. All right, so Bacillus subtilis is a good bacteria. Now, what happens is it will, so it will start to essentially fight for the plant. It will start to it will start ramping up its population, the bacillus. It'll start colonizing the root area more. It'll start producing chemicals that can fight off pathogens in the soil. But the other thing that it does is it tells the plant, close your stomata. So the activity of the bacillus is feeding back to the plant. The plant is closing its stomata in response, and that helps to keep Pseudomonas more at bay. So basically, microbes in the soil are telling the plant, hey, you got Pseudomonas on you, you better shut your stomata. Essentially, that's the upshot of it. And so this has been nicely documented in a very nice study. They're also showing this other idea that you have Alternaria. They're showing Alternaria solani is a fungus that can also attack tomato. And they're showing that in this case, they're showing a nearby tomato plant, and they're showing Glomus mossae. What do you think that says, this Glomus species? What are they trying to demonstrate here? It's written down. Yeah, that's the mycorrhiza. So they're showing through this mycorrhizal bridge that you can have chemical communication from this plant that's getting attacked. And it's warning its neighbor, you're next, you better ramp up. And so they're showing this dynamic where soil bacteria are telling the plant getting attacked at its leaves, close your stomata. And then the mycorrhizae helping to send the signal over to a nearby plant, your next, ramp it up.
Is there something within the tomato plant that the Pseudomonas just likes? Is it like a chemical compound that?
Yeah, I guess, so I talk about this in Plant Fizz. In fact, I'll be getting into this in like next week, but basically certain pathogens are just really well adapted to attack certain plants. And Pseudomonas, I guess, with this idea of coevolution, they call it the evolutionary arms race. The arms race in this idea, they say the arms race because it's a pathogen trying to attack the plant trying to defend itself against the pathogen. So it's this co-evolutionary dynamic where Pseudomonas is just pretty effective at attacking tomato. And Pseudomonas, though, that can attack a lot of different plants, but tomatoes impacted by, I don't know. I think it's over 100 different pathogens it's susceptible to. So if you're a tomato grower, hats off, because that's a tough one.
Another question.
Yeah.
Do you think that that's due to how we've like domesticated the plant?
Yes. So there are some intriguing studies that, not on purpose, but when we breed plants for food production, we didn't really give a lot of thought to plant defense ideas. So this is through selective breeding ideas that have been going on for centuries. The idea that, you want a certain sweetness to a food, sweet corn, for example, is so super sweet now compared to the wild relative we bred it from. It's amazing. And so, but it sells, right? If you can sell sweet corn, people buy it. And that's, our love for sweetness. Well, in that process, though, they've kind of bred out some of the plant defense. They didn't do it on purpose. It just, when they were doing selective breeding for, I want really sweet corn, some of the plant defense mechanisms went to the wayside. And so this is an area, there's a lot of scientists who say we should go back into the wild relatives and like try again and try to like rebreed some different crop species that have better capability of defending themselves. So yeah, that's a problem that was a consequence of trying to breed certain traits into the plant that humans like. So, but that was back in the era when we just didn't understand how pathogens work and the dynamics of it. So really, really interesting ideas out there when you look at how the microbes are influencing the ability of the plant to defend itself. This is also a really interesting idea. This is coming from kind of taking the idea of how microbes hang out with humans and the human microbiome and saying, I wonder if there's an analogous situation in the plant world. And so when you think about especially the plant roots, so deeply colonized by so many microbes, they are influencing the plant strongly, we now know. And so they started talking about this idea of soil memory, and there's been a little bit published about this. It's a really good concept. I think there's some interesting ideas here. So soil memory as a potential mechanism for encouraging sustainable plant health and productivity. So Jorge Vivanco is a long-term researcher in plant microbe interactions. And so his lab has been looking at this idea. And so these are some of the quotes from his paper and some of the conclusions, and looking at how you have these microbial partnerships in the soil. And he's saying it's much like the mammalian microbial colonizers that are passed off to their offspring. So when you were born, you were immediately colonized by microbes from your mom during the labor process. Absolutely. And then, of course, as the caregiver, her microbes on her skin and in the breast milk all influenced you strongly, as well as then anyone else in your household, of course, any animals in, pets that you have, all of that is influencing you. So this idea of, getting the maintenance of the rhizosphere community of microbes is similar to the development of the human microbiota. Colonization begins at birth, so when the seed is germinating, and as the infant matures, the seed is becoming a seedling and a young plant. The microbial community increases in population size and complexity, seeded by intimate contact with caregivers and the environment. Think about pollinators for perhaps, or other organisms that come by and interact with the plant. Microbes acquired by individuals to successfully adapt to their environment are then passed down to their offspring. We already know that seeds, for example, contain endophytes and epiphytes. And the idea now is, do we really need to surface sterilize those seeds? Is that something we should worry about? Maybe that's important microbial partners for their offspring. We document that the current literature displays evidence that a similar phenomenon may occur in plants through a kind of soil memory. And so they call it the soil immune response. So what they are trying to show here is, here you have time of cultivation, and then disease severity on the y-axis. So what they're trying to show here is that you have a scenario where a pathogen comes into the system. So first entry. And they're saying this is through a root infection, so it's a soil pathogen. And you end up with a severe disease outbreak. And they're showing this uptick here in disease severity, okay? And then they're showing this idea that when they say conducive, they're saying it's conducive. That's a fancy word that they use in plant pathology to mean that the plant is susceptible to the pathogen. Okay, so it's conducive conditions for the pathogen. And so they're trying to show that under conducive conditions, you have this pie chart here showing low diversity of microbes hanging out with the plant. Okay, so they're by the different colors there, and not many different colors. They're just trying to show, there's not a lot of diversity there in the microbes that are hanging out with the plant. Then, if you continue on with this, what's happening is, over time, if the plant is surviving this pathogen attack, what's happening is that there's enrichment where the microbes in that soil are being recruited, and the microbes that are persisting, the plant is actively recruiting. They are helping fight off the pathogen for the plant. And so you start to see this decrease in disease severity. So the plant survived the initial pathogen attack by recruiting key microbes and ramping up diversity. You can see here suppressive. That's another fancy word they use a lot in plant pathology. If something is suppressive, if a soil, we know that there's certain soils that we call suppressive soils that have been heavily studied. And when we say that, they're suppressive to a certain disease that used to be there, and now it's no longer there. The disease is gone. And so we say it's a suppressive soil. So when you go and examine these soils, often what you find is really high populations of beneficial microbes to the plant and really good diversity, lots and lots of microbes. And so that's what they're trying to show. And then they're showing that the disease severity goes down. And then what they did is they hit the plants with that same pathogen again. Okay. So after the plant recovered for a while, then they said, okay, let's expose the plant to that same pathogen, put it into the soil and see what happens. And you can see that the plant really didn't get any severe disease outbreak. So it's very much like your immune system, your memory T cells do that for you. So once you've been exposed to a certain pathogen and you made it through that, very often your body is busy making memory T cells so that if you get exposed to that same pathogen, your body goes, oh, yep, yep, I know that one. And your memory T cells are there. Boom, you get that pathogen wiped out. You probably don't even know you have it in you because you don't feel sick. And so it's kind of that same idea that there's soil memory of that pathogen in the sense of the microbes that now are with that plant have shifted. They're the microbes that can really help the plant deal with that same pathogen. So the pathogens are, I mean, the beneficial microbes in this suppressive condition are kind of like the memory T cells. If you want to use that analogy, that's a fair way to think about it. Isn't that cool? So this is just really exciting ideas. I mean, with suppressive soils, we do see that. I mean, and it's so cool that they've gone back to these suppressive soil ideas and started thinking about, okay, can we use this knowledge to really ramp up plant defenses against these key pathogens? And what I thought was great is that they're thinking about this high diversity idea, and that you really need to look at the community of microbes with that plant. So back early days of biocontrol experiments, what they would do is they'd say, well, this microbe can really inhibit this pathogen. And so they'd put this one microbe into the soil and say, please kill that pathogen. And often it wouldn't work because it's just one microbe against all these others. And so they're realizing that's not really the best strategy. If you want to try to help protect the plant against pathogens, you need a suite, a consortium, a team of microbes that can do this, and through quorum sensing, so that they can, as a community, help that plant, good diversity of the microbiome. And as you know, that's the general rule of thumb. The greater the diversity of an ecosystem, the more healthy it tends to be. So this community perspective. And so they're looking at mixtures of different microbial species. And again, they need to work as a team. They're looking at quorum sensing for this idea, or nanotubes, or the extracellular vesicles, something that they can easily communicate with each other. You don't need to know the word syncoms. That's not necessary. But basically this idea of we need to put in mixtures of beneficials. And the other idea too is to look at maybe using the plant, breeding the plant to release key nutrients to attract these good microbes, this community of good microbes. Augmenting the indigenous microbes that are already there, absolutely. So work with what you got because they're already there in the soil. They've adapted to that soil. That's a very good strategy, rather than if you try to spike in a microbe that's not from that habitat, that's challenging. So nowadays, when you go looking for biocontrol agents, you go to the diseased soil and you root around for what's in here that can inhibit, rather than trying to find some random microbe in some other strategy. You go to the site that's the problem and try to find the microbes there that can help the plant. Breeding again for the plant, looking at that. So they're trying to come at it from a lot of different angles. They're realizing putting one biocontrol agent into the field is not effective. You need many angles of approach. And so basically this is the idea of having these really good beneficial microbes there. It's kind of like taking a prebiotic where you're helping support the good microbes in the soil, making sure that they're well fed, get the plant feeding that through their excretions from their root, and doing what you can to augment the soil to keep those good microbes there. So they're really starting to get more sophisticated in how they think about using microbes, and that it's important that we take many different avenues of attack against plant pathogens, and just helping the plant stay healthy and support its immune system. And then this other idea also recently published, well, fairly recently. I think we'll stop there. I'll talk about, there's a couple other things I want to talk about, and then, so we'll wrap this up tomorrow. and then get into the next topic of plant animal interactions.